Okay, and let's get started. So welcome everyone. It is time for another edition of National Journal Club. Very excited today. The folks at UT Southwestern will be presenting, led by Dr. James Cow, and his fellows, Dr. Schaefer, Meadors, and Dr. Weiss. And I think there's a guest star appearance by Dr. Paul Wax. So watch out. Today they will be covering novel antidotes in medical toxicology, and this is going to be a great discussion. Just some usual housekeeping. Remember, if you're out there in the Twitter sphere, use the hashtag ACMTNJC. We will break between articles, and please raise your hand, and we'll be happy to acknowledge you and get you in the conversation. And with that, James, you want to take it away?
Yeah, thanks, Steve, for the introduction. I'm here with my three fellows. I think the overall introduction is going to be fairly short because each of these articles for these antidotes kind of stand on their own. So I'm going to let my fellows take it away from here and start. We'll talk about the colchicine article first.
Yeah, so my name is Sarah Schaefer. I'll be presenting the colchicine article, but first I'd like a shout out to the house band for providing us with that wonderful music before we got started. So the title of this paper is Anti-Colchicine Fab Fragments Prevent Lethal Colchicine Toxicity in a Porcine Model, or colchicine-flavored bacon, as I like to call it. And so this was a translational study, but before we get started into the actual article discussion, we're going to ask a few questions for the audience. So first of all, we'd like to open up the polls to ask if anyone has ever managed a sick colchicine ingestion, either in person or through the Poison Center.
So the poll is open. Please answer.
The poll is also online on Twitter for those of you following along.
Great. So both WebEx and Twitter. Okay. Well, it looks like we had 7 out of 9 answer, but it looks like everyone that did answer has indeed managed the sick colchicine ingestion.
Great. So we'll move on to the next question. The next question is a question about the timing of presentation. And so our next question is, how many hours post-exposure did patients present to the healthcare facility? And this can just be a rough estimate, of course.
Great. And folks, that poll is open now too. Please weigh in. Okay, it looks like we got 7 out of 10 answers here. And if
Steve,
you can,
what do the answers look like? We can't actually see the answers. I'm curious what the timing has been for people.
Sure. So 10%, 1 out of 10 said less than 1 hour. 30% said 1 to 3 hours. No one was between 3 and 6 hours, and 3 out of 10 greater than 6 hours.
Okay. Great. So this will become relevant later on in our discussion. And so, you know, I'm going to just briefly touch on colchicine. I think most of us are familiar with it. It's derived from the autumn crocus plant, and we know that it's a microtubule poisoner. So what it does is microtubules are made up of these alpha and beta tubulin proteins that form a heterodimer. And so colchicine binds at the colchicine binding site on the heterodimer. And so what happens is it prevents polymerization. But at high enough concentrations, it may actually even promote depolymerization of the microtubules, which then causes cellular dysfunction. And so colchicine is very toxic in overdose. And so the number we throw around is that survival is typical in doses of less than 0.5 mg per kg. Death is typical in doses of greater than 0.8 mg per kg, which is a pretty narrow therapeutic window. And of course, deaths have been documented at less than 0.5 mg per kg, and survival has been documented at doses over 0.8 mg per kg. But this just gives you an idea as to the toxicity of colchicine and overdose. So this study is a translational study, like I mentioned earlier, and what they're trying to do is they're trying to study the efficacy of ovine-derived colchicine Fab. And so Fab fragments are derived from antibodies where the Fc fragment has been cleaved at the bottom, and so you're left with the Fab fragments that will bind to whatever your target molecule is. And the thought is that this helps prevent the the sort of allergic reactions that we can see with immunoglobulin therapy. And so we commonly use Fab fragments for crofab for crotolid snake bites and also in digoxin toxicity. And so for this study, they used ovine-derived Fab that was used from microfarm laboratories, and so they used a dose of about 12.5 mils per kg to neutralize a colchicine dose of 0.25 mg per kg. And so this will kind of come into play later when we talk a little bit about what that dosage actually means. And so the study actually had two phases. So the first phase of the study, what the researchers were trying to do is they're trying to come up with a model of colchicine toxicity where they could give these mini pigs a dose of colchicine that would cause sort of a death that would mimic what we see in human toxicity, so multi-organ failure and cardiotoxicity, but they wanted survival to last at least 24 hours because they didn't want these pigs to die right away. And so the second phase of the study then is to study the pharmacokinetics and the effects of the anti-colchicine Fab to try to show that they did have pharmacologic efficacy when they gave it to the mini pigs to show that it would have application in a biologic model. And so the overall goal of the study for them is to try to prove that this anti-colchicine Fab works so that then they can use it for human studies and try to study it to see if it would be an effective antidote. So for the first phase of the study, they originally were trying to study the pigs with an oral dose of colchicine because people take oral colchicine, so this was thought to be a more realistic model for human poisoning. So they initially chose a dose of 0.5 mg per kg to try to get a dose that was high enough to cause toxicity but not guarantee death. And then they chose a 1.0 mg per kg oral colchicine model to get a higher dose to cause toxicity and death. But what they found when they tried this oral colchicine model was that they had really sort of variable clinical responses. And so there were 4 pigs in the study, so the lines in the graph actually represent the 4 different individual pigs. And the 0.5 mg per kg doses in blue and the 1.0 mg per kg doses in red. And so you can see that the endpoints are sort of all over the graph. And even the time to euthanasia was not dose dependent. And so the researchers felt like the oral model of colchicine toxicity was not reliable enough to use as a model to test the anti-colchicine Fab. So then they went back to the drawing board. This is more parameters to kind of show that the endpoints were sort of all over the place, not quite dose dependent, and didn't really provide for a reliable model. So they went back to the drawing board and decided that they would use an IV bolus colchicine dose. They thought maybe there was something to do with the anesthetized pigs that they weren't absorbing the colchicine from the gut as reliably as humans might. And so they chose the IV bolus colchicine dosing. And so instead of a 0.5 mg per kg, they actually went down to a 0.25 mg per kg IV dose. And their reasoning for doing that was to try to make up for the increased bioavailability of the IV dose. We know that oral colchicine has a bioavailability about 25 to 50%, depending on certain studies. And they kept the 1.0 mg per kg IV dose for the colchicine because they wanted to have a higher toxicity group, sort of as a point of comparison. But what they found with the models is that the 1.0 mg per kg IV bolus group, it died too soon. The pig actually died within 14 hours, so it didn't reach that sort of 24-hour benchmark they were looking for. And so what they ended up doing is they chose the 0.25 mg per kg IV colchicine group as their model for toxicity so that they can then test the anti-colchicine Fab. So now that they have this model for toxicity, they found that the 0.25 group actually had pretty good pharmacokinetics and that most of the pigs were within range that they felt like was reliable enough to be a model for their testing. So then they went on to test the anti-colchicine Fab. So they used the 3 pigs from the original toxicity model as a control group of sorts for the rest of the pigs. And so they had 3 different groups of pigs that received the anti-colchicine Fab. Each group contained 2 pigs, so we're talking about 6 pigs total that received anti-colchicine Fab. So the first group received Fab 6 hours after they finished their colchicine infusion. So what they did is they started the study, they infused the 0.25 mg/kg dose of colchicine over an hour. They thought that this might approximate the absorption from the gut. And so after that hour infusion was over, they waited 6 hours. And the 6-hour group is interesting because they gave half of the neutralizing dose of Fab over an hour, and then they gave the other half over 6 hours. The reasoning being that they wanted to prevent the free Fab that was wasted in urine. But then the 3-hour and 1-hour group had a completely different administration of the anti-colchicine Fab. So the 3-hour group got the total neutralizing dose of Fab over an hour, 3 hours after they finished the colchicine poisoning, so 4 hours into the study. And then the 1-hour group got the full dose of anti-colchicine Fab 2 hours into the study. And what they found was that this chart here kind of summarizes the pharmacokinetics for the Fab itself. It's a confusing chart because you have the 4 groups with the different colors corresponding to the different groups. But the circles represent the free colchicine, which is different than the colchicine that was actually bound to the Fab. And so you can see for the control group, it had an initial peak within the first few hours of toxicity, and that's the blue color, and then it quickly drops to a really low free colchicine level. And then the Fab from 6-hour group is the red graph, and you can see that about 6 hours into the study, 7 hours into the study when they received the colchicine, you see the rise in plasma colchicine concentrations that corresponds with the colchicine binding to the Fab in the serum and giving us these higher plasma levels. And the green group is the Fab from 3 hours, and the purple group, which is the triangle, you can see that it's the highest peak here is the 1-hour group. And so what they found was the 6-hour group had a 7.35-fold increase in plasma colchicine and removal of all the free colchicine, but it didn't actually increase urinary excretion. And we'll look at some graphs later. It showed it did not actually prevent toxicity or improve outcome, and those pigs were euthanized at about 29 hours. And so they euthanized the pigs essentially when they stopped responding to pressors and developed severe cardiotoxicity with a MAP less than 45. The 3-hour group had a 6-fold increase in plasma colchicine and a 2.1-fold increase in urinary colchicine elimination. And the 1-hour group actually had a 12.9-fold increase in plasma colchicine and a 2.5-fold increase in urinary colchicine elimination. And we'll look at a graph later that shows that these pigs actually were euthanized after the end of the study because they survived until the end of the study period, which was, the study period was, I believe, 48 hours. Yeah. So this graph shows the time to euthanasia in the animals, and so you can see the 1 mg/kg. This was the pig who got the IV dose that it died relatively quickly, and the 0.25 mg/kg group, whether the control group versus the Fab from 6 hours actually died before the end of the study, but both the 3-hour and 1-hour groups survived to the end of the study and were euthanized afterwards. These are just some charts that show the various groups and the biochemistry during the study that they checked, so things like urea, creatinine kinase, creatinine, and the LDH. And I think the biggest takeaway here is you can see that the blue and the red line, so the blue line is the control that got no Fab, and the red line is the 6-hour group, seem to trend together, versus the Fab from the 3-hour or 1-hour group actually tend to follow each other. And so this next slide shows similar parameters for albumin, AST, and ALK phos. And so you can kind of see a similar trend there. So figure 6 shows blood gas analysis in the different groups, and it's similar to the charts we just saw. So the control group is blue, the FAB group, the 6-hour FAB group is in red, and the purple and green represent the 3 and 1 hour groups, and so you can kind of see a similar trend there. What's interesting is that the 6-hour FAB group actually had a worse pH compared to the control group. And then in the lactate, you can see too, the control group and the FAB, the group that got FAB at 6 hours actually start to have an increase in arterial lactate. But they did note in the paper that there was one pig in the 1 hour group that had a transient rise in lactate to 5.0 that was unexplained. And because each group has 2 pigs, it's hard to know what the significance of that is. Is that pig an outlier, or do we have to worry about some kind of elevated lactate after colchicine FAB administration? This last one is just the venous blood glucose. So overall, the conclusions of the study showed, so remember the phase 1 was about the IV versus oral model of toxicity. So the oral colchicine dosing does not provide a reliable model of toxicity according to this study. They found that if you can give anti-colchicine FAB early, then it redistributes colchicine back into the serum and it increases urinary elimination, preventing toxicity if the dose is given sufficiently early and if it's given in a sufficient dose. And so we kind of talked about how you need a large dose of the anti-colchicine FAB to actually reverse the colchicine. So again, the study required a 12.5 mil per kg to fully neutralize a 0.25 mil per kg dose. So that meant in a 30 kg pig, the pig required 375 mils of colchicine. And so that can be a fairly large dose of a FAB product. And so the question is, how does this translate into a human model? And would this large dose of colchicine FAB potentially cause increased problems with anaphylaxis and serum sickness? Another thing to acknowledge as well is that human serum levels of colchicine, so there's a study that was done, it gave humans a one-time 1 milligram oral dose of colchicine that resulted in a 3,000 nanogram per mil serum or plasma level, where the pigs in the study had about a 200 to 300 nanogram per mil plasma level after they got the IV dose of 0.25 mil per kg. So the pig model may not be as reliable for a human model for toxicity. And I think the question is, does this change in plasma colchicine translate to maybe an increased need for anti-colchicine FAB dosing compared to the pig model, since their plasma levels were only 200 to 300 compared to 3,000 for humans. And then I think there's a problem too, the anti-colchicine FAB needed to be given within 1 to 3 hours of poisoning based on this study. And patients who got the 6-hour FAB actually did a little bit worse depending on how you interpret the data, and at the very least did no better than the controls. And so, you know, the question is, are patients going to fall under, human patients going to fall under the same time constraints as the pigs in this study actually did? You know, if patients come in early enough, will FAB be effective? Do we have to worry about patients that present after that 3-hour mark? And I think that's why we will really have to be careful when we look at how the anti-colchicine effect translates into humans. So the conclusions of this paper is that clinical trials are required to translate FAB effects for human patients. And I think, you know, we all are excited to see whether colchicine FAB may be an effective therapy. You know, we do use FAB in various antidotes, but there have been many studies in the past that showed promising animal data regarding a FAB antidote that didn't actually translate into clinical practice. So I touched on this in the conclusion slide, but some of the limitations of this study are the FAB dosing. You know, are patients going to, human patients require a massive dosing of FAB, and is that going to set them up for increased adverse effects related to that FAB? The study design itself had a lack of blinding, and there was a lack of control in the study design. So they used the pigs from the poisoning model as a sort of control, but there was no control group where the pigs actually just received the equivalent fluid dose in the FAB, but actually did not receive FAB. Also, the problem is the route of colchicine administration. So humans receive mostly oral dosing, and so the question is, does IV pig dosing given over an hour really approximate what happens with humans when they overdose on colchicine who are going to overdose on a PO pill. And then the timing needed for Fab effectiveness is probably the biggest limitation to the potential for anti-colchicine Fab and its use as an antidote in the future. So that's the presentation for this anti-colchicine Fab paper. So if anybody has any questions, feel free to chime in and let us know if you have any comments, thoughts, questions.
Yeah, and again, for folks out there, please raise your hand if you'd like to make a comment or ask a question. That would be great. Wonderful review. Really, it's a fascinating paper, and I think just that time frame, that within the first 1 to 3 hours, that seems to be when, when it's critical to give it. I know the French have done some work with this, and I don't recall the details. Do you remember in their experience, was there an effect like that, where if given earlier, there's a better outcome?
I know that right now they're doing human clinical studies in France. I don't actually know the outcome of that data. There was a case report where a patient got the Fab 40 hours after ingestion, and they had a good clinical outcome. And I think what's interesting, too, is that some of these pharmacokinetic studies in humans showed that some humans had a peak in plasma colchicine levels at 2 hours, but other patients also had a peak at 12 hours. So there's a question as to whether individual drug elimination may play a role in whether Fab's effective. And then also, colchicine goes through the enterohepatic recirculation, so I don't know how much that's going to play a role, too, and whether humans are going to respond to the anti-colchicine Fab in a real-world clinical setting later than that sort of 1 to 3 hour cutoff.
Right. No, those are good ones. Yeah, go ahead.
Yeah, just reviewing some kind of a review article that was published in 2010. There was just a report of a single human case report of using Fab that may have been efficacious. And then there's some in vitro studies that shows it restores tubulin activity. But beyond that, this is pretty limited, and this is kind of a new avenue that they're going on with this product.
Right, right. So really, we would need clinical trials to see if this will get us anywhere.
Yeah.
All right, great. And not seeing any hands. Any other comments on this? All right, I don't see any hands. So why don't we press on to the next paper?
Okay, I am Kevin Beth Metters. I'm a junior fellow here in Dallas, and I'm presenting idarucizumab for dabigatran reversal. This is the 2015 interim analysis paper. So for my audience response question, in your hospital, how would you reverse bleeding in a patient who takes or overdosed on dabigatran? Would you use dialysis, factor VIII inhibitor bypassing activity, FFP, idarucizumab, PCC, or recombinant activated factor VII?
All right, the poll is open. Please go for it, everyone. All right, it looks like we have 8 out of 12 responded. Let me read you the results here. So we have actually, so 4 didn't answer. Actually, everyone that answered will use the antidote. And how do you pronounce that?
I know, I've practiced. I say idarucizumab. Like
I dare you. I like that. I dare you. Okay.
Yeah, that's what I'm sticking with. Good. I'm glad to know that most of our audience would use the antidote that I'm going to be discussing here. So just a little bit of background. Historically, vitamin K antagonists were the only option for anticoagulation, but the NOACs, the new oral anticoagulants, were developed to require less frequent monitoring and less patient variability. The one I'll discuss today is the direct thrombin inhibitor, dabigatran. And there are also direct factor Xa inhibitors, such as rivaroxaban, apixaban, and edoxaban. The reversal agents are limited though, and that's one of the big major roadblocks for using these medications. So dabigatran, again, is an oral thrombin inhibitor. It's licensed for the prevention of stroke in patients with non-valvular AFib and for the prevention and treatment of venous thromboembolism. It is associated with less serious bleeding than warfarin, but it can have life-threatening bleeding, and it can increase the risk of perioperative bleeding. Idarucizumab is a monoclonal antibody fragment that we'll discuss. It binds both free and thrombin-bound dabigatran with 350 times the affinity of that of thrombin. As you can see in the graphic here, dabigatran is the little guy in the middle who would usually bind to thrombin, but when idarucizumab is present, it will preferentially bind idarucizumab, leaving thrombin unbound. This paper discusses briefly a previous study that showed promising results, which prompted our authors to research it further. Oh, one thing I wanted to mention here, you may have noticed the Praxbind logo and the Pradaxa logo are very similar. That's not by coincidence. The creators of Pradaxa, which is dabigatran, also created the idarucizumab Praxbind. So the methods. This is an ongoing multi-center prospective cohort study, or rather it was when it was published in 2015. We'll briefly touch on the full cohort analysis that was published in 2017 at the end. Each patient was divided into either Group A or Group B. Patients in Group A had overt, uncontrollable, or life-threatening bleeds, and patients in Group B required emergent surgery or an invasive procedure that couldn't wait 8 hours until dabigatran could have been out of their system on its own. So again, the plan at this time was to recruit up to 300 patients at more than 400 centers in 38 countries. They published the results of the first 90 patients that were broken up into these two groups. All patients were greater than 18 years old and taking dabigatran. So all of the patients received 5 grams of idarucizumab IV in 2 milliliter bolus infusions, which were 2.5 grams each, no more than 15 minutes apart. That 5-gram dose was calculated to reverse the total body load of dabigatran that was associated with the 99th percentile of the dabigatran levels measured in the RELY trial, and that was in 2014. Blood samples were obtained at baseline, and then after the first infusion of idarucizumab, and then between 10 to 30 minutes after the second, and at 1, 2, 4, 12, and 24 hours after the second infusion of idarucizumab. The primary endpoint that they looked at here is the maximum percentage reversal of the anticoagulant effect of dabigatran using 2 clotting factors, the dilute thrombin time and the Ekerin clotting time. The equation that they used is shown below. Calculated values of 100% or higher were interpreted as complete reversal. The secondary endpoints were mainly clinical outcomes, and these include restoration of hemostasis, the decrease in concentration of dabigatran, the number of deaths, thrombotic events, and other serious adverse events. And these were assessed by treating physicians. So we'll get right to the results. This table looks at the demographics. Again, there's 90 patients total in this interim analysis. 51 were in Group A and 39 were in Group B. The median age in years was 76.5. The median time since the last dabigatran dose was 15.4 hours. The median creatinine clearance was 58 mils per min, and the percentage of patients who were taking their dabigatran for atrial fibrillation was 96%. And you can see in Group A and Group B, the values are not too different. All 90 of these patients were enrolled from June 2014 through February 2015 at 184 sites in 35 countries. So the primary endpoint results are here. All of the 90 patients who were enrolled, everyone received the dilute thrombin time and the Ekerin clotting time at baseline. 22 of those patients actually had a normal dilute thrombin time at baseline, which means only 68 of them were included in the efficacy analysis. And similarly, with the Ekerin clotting time, 9 of the patients had a normal clotting time at baseline, so only 81 out of those 90 were included in the efficacy analysis. The median maximum percentage reversal with idarucizumab by means of both clotting time measures was 100%, and that's a 95% confidence interval from 100 to 100. This graph looks at the dilute thrombin time after idarucizumab administration. As you can see at baseline, the thrombin time was, let's see, it was normalized in 98% of the patients in Group A, and it was normalized in 93% of the patients in Group B. So you can see here, this is the baseline level is the first one shown, and then after the first infusion, and then 10 to 30 minutes after the second one, and every other hour increment, it was significantly reduced. The ECR clotting time showed similar results. It was normalized in 89% of the patients in Group A and 88% of the patients in Group B. And then similar results were observed with respect to the APTT, the activated partial thromboplastin time, and the thrombin time that were measured at a central laboratory. This graph looks at the drop in initial dabigatran concentration. So as you can see, there were levels of dabigatran in these patients because, again, all of them were taking dabigatran at baseline. So the median plasma concentration of total dabigatran was 132 nanograms per milliliters in Group A and 114 nanograms per milliliters in Group B. And then this graph actually looks at the unbound dabigatran. In Group A, the median plasma concentration of the unbound was 84 nanograms per milliliter, and in Group B it was 76 nanograms per milliliter. After the first vial of idarucizumab that was administered, the concentration of the unbound dabigatran decreased to less than 20 nanograms per milliliter in all but one patient, and you can see that remains low throughout the blood samples. Idarucizumab pharmacokinetics are shown here. Again, there was no idarucizumab in any of the patients at baseline because these patients were taking dabigatran. But then after the first infusion, they did show a rise in the concentration of idarucizumab, as would be expected. And then 10 to 30 minutes after the second infusion is when the peak concentration of idarucizumab was seen in both groups. By 4 hours after administration of idarucizumab, the mean plasma concentration of idarucizumab had been decreased by 80% from the peak level. This graph shows another one of the secondary outcomes, which was time to bleeding cessation. In Group A, there were 51 patients. 3 of the patients did not receive a bleeding assessment at baseline, and 13 of the patients, the time to cessation of bleeding could not be obtained. So out of the remaining 35 patients, the time to cessation of bleeding was 11.4 hours, and that's a median level. That's a little, it's an interesting statistic because when you consider a lot of the different groups, like the patients with non-intracranial hemorrhage or GI bleeds, the time to cessation of bleeding was drastically different. The time to cessation of bleeding was only 2.5 hours among patients with non-ICH, and then it was 3.5 hours among patients with GI bleed, and then 4.5 hours among patients with no intracranial hemorrhage or GI bleed. And again, it was 11.4 hours when you considered all the patients that the time to cessation of bleeding could be obtained. In Group B, we looked at operative hemostasis outcomes. There were 39 patients total, 1 of which no longer needed the procedure after the antidote. 2 were still unstable for surgery. So 36 patients underwent procedures in the end of the study, 33 of which is 92%, had normal intraoperative hemostasis, 2 had mildly abnormal hemostasis, and 1 had moderately abnormal hemostasis. There were 21 patients who had serious adverse events at the end of the study, 18 deaths, 9 from each group, and 3 in Group A from acute bleeding. 5 patients had thrombotic events, 2 had GI bleeds, 1 each had postoperative wound infection, right ventricular failure, delirium, and pulmonary edema. And as you can see, some of the patients had more than 1 serious adverse event. Of the 5 thrombotic events, they were broken up into 2 categories, early and late. The early thrombotic events were within 72 hours after the antidote administration, and the late ones were more than 72 hours after the idarucizumab administration. Only one patient was within the early time frame, and thrombotic events during that time frame were thought to be more likely related to the antidotes. However, antithrombic therapy had not been reinitiated in that one patient. So for our discussion, idarucizumab rapidly and completely reversed the anticoagulant effect of dabigatran in 88 to 98% of patients that had elevated clotting times at baseline. It obviated the need for intervention in 1 out of the 3 Group B patients who did not undergo their procedure, and it produced normal hemostasis in 92% of the Group B patients who did undergo their procedure. It only had 1 of the 90 patients who had a thrombotic event within 72 hours after the administration. The paper did have limitations. It was not controlled. It was industry-sponsored. They didn't discuss FFP or PCC as alternative treatments or treatments simultaneous with the idarucizumab, and it failed to correlate it to the APTT value. This is probably one of the only values that we can actually get in the emergency department in real time, so that may have been helpful. And then again, the time to cessation of bleeding showed very different measurements when you considered patients with different adverse events. But my conclusion is that idarucizumab rapidly and safely reversed the anticoagulant effect of dabigatran. And again, I'll briefly touch on that full cohort analysis that was published 2 years later in 2017. It actually looked at 503 patients. And again, it also showed that the median maximum percentage reversal of dabigatran was 100%. Here, when they included all the patients, including those with intracranial hemorrhage, the time to cessation of bleeding was only 2.5 hours, as opposed to our paper, which showed 11.4 hours. So you can see that was very different, but again, the primary endpoint that they looked at was the same, with the reversal being 100%. And I'll take any questions or discussion points.
All right, that was an awesome summary. And again, please, audience out there, raise your hands. We'd love to hear from you. Yeah, I guess bottom line, it seems like all of our folks with the poll said that they would reach for this first. I'm trying to come up with a counter argument to not do that. It seems difficult.
Yeah, so Steve, just one other comment from me is that one of the reasons why they said this was not a controlled study was they said it was unethical to not give this antidote because there were no alternative antidotes. And we do have some studies out there for like FFP, PCC, dialysis. There are other ways to manage this. I don't know if they're ideal or not, but it would have been great to have a head-to-head comparison of standard care without this versus giving the antidote, right?
I totally agree, yeah. Yeah, and these overdoses or the bleeding from these, from dabigatran, it's tricky because there's no convenient way to measure the degree of anticoagulation. Right.
Yeah, and that's one of the Twitter polls I just put out. Does anyone have a DTT or ECT available out there to measure, to follow this? So, don't know.
That's right. Okay. All right. I don't see any hands out there. Yeah, no, it is just interesting from our perspective right now. We are swimming in the brodifacoum-tainted synthetic cannabinoids. And, at least, quite grateful that at least we have an INR that really tells us what we're doing and guides our vitamin K therapy. It would be great if these novel agents, if we had a better way to measure the degree of anticoagulation.
Yep, definitely agree.
All right, so without seeing hands, why don't we press forward?
Okay, great. This is Stephanie Weiss, and I am going to be presenting the third paper, which is a little bit different than the kinds of things that we normally talk about during this journal club. This is actually a basic science paper looking at the use of nanoparticles as a potential antidote for envenomation. So there's basically two potential ways that you could use a nanoparticle. In the top one there, you're seeing something that's more along the lines of using the nanoparticles as a sort of scaffold to use ligands like antibodies that are very specific for the antigen under question. That's not what the authors were doing in this particular article. What they were interested in doing is to generate polymers that have intrinsic affinity and selectivity to a range of toxins. And so here on the bottom, what you're seeing is that it's a capture agent, that the polymer is a hydrogel that's formulated with functional groups that are complementary to target protein domains. And so what they were doing was using an iterative process to improve the affinity of the nanoparticle for the target peptide, and they do that by optimizing the composition and the proportion of the monomers that they're using. So since these nanoparticles are being formed by a kinetically driven process, the sequence of the monomers is actually not being controlled. It's only an average composition, and again, they adjust that by changing the stoichiometry of the monomers that they put into the feed. And then they do some cross-linking as well, and I'm going to talk a little bit more about that in just a minute. So what you're seeing here is that some pros and cons basically for nanoparticles versus antibodies. The biggest issue with the nanoparticles, or one of the biggest issues with them, is, as I mentioned, they are nonspecific, and you can have nonspecific binding to other proteins as opposed to antibodies, which are developed to be extremely specific for their protein targets. But on the plus side, the nanoparticles are much more chemically stable. They are enzyme resistant. They have a much higher binding capacity. They're much cheaper and faster to synthesize. And then the authors at least believe that there's less risk of immunogenicity, although of course that's potentially up for question. And here what you see are some of the monomers that were used by these authors to create the polymers that they made. And you can see they're all basically acrylamide based. Some of them are hydrophobic, some are hydrophilic, some are charged positively, some are charged negatively. And then on the left-hand side in the middle, the bis one is actually the linker that they used. On the bottom right, you can see an example of what the polymers look like. So it's a chain that's kind of somewhat resembling what a peptide chain looks like, and you can see that the functional groups from the different acrylamide monomers are hanging off that. And this slide basically shows in general how they produce these nanoparticles. So as I mentioned, they're basically taking a pool of these monomers, and they're trying to synthesize a library of nanoparticles using free radical polymerization, and they then purify these by dialysis. So as I mentioned, the side chains are distributed randomly, but since all of these monomers are acrylamide-based, then they have similar reactivity. And so even though the incorporation of each exact monomer is random, they do have similar proportions based upon the proportions of the monomers that are used. So one of the things that's distinct about these compared to peptides has to do with the fact that the backbone of this, when you make the polymer, it is all sp3 hybridized, as opposed to a peptide, which is the amide bonds in the peptide have partial sp2 hybridization, and so they're therefore much more rigid compared to the nanoparticles here. So they do some cross-linking, as I mentioned earlier, in order to help account for that and get a little bit improved rigidity for their own particles. The N-isopropyl one, which is at the very bottom on the left-hand side, that monomer was chosen as their backbone just because there's not very much innate biomacromolecule affinity. In panel C, what you see is an electron micrograph of what these polymers look like, and they're about 50 nanometers in diameter, which is about 100 times smaller than a red blood cell, and it's about the size of an IgM molecule. Other goals that they have for making these, they want them to be stable and inert and non-toxic. And they also talk quite a bit about wanting to have them stay in the bloodstream long enough to capture the target venom molecules. So if they're too small, if they're less than about 8 nanometers, then the kidney will clear them. And if they're too large, so greater than about 200 nanometers, then they will basically be sequestered by the mononuclear phagocytic system in the liver and the spleen. So that 50 nanometer diameter is kind of in that sweet spot in between them. And the other thing they mentioned too is that if they're strongly charged, those particles also get phagocytosed more readily compared to neutral nanoparticles. So when they use nanoparticles in vivo, one of the things that happens is that there's a bunch of proteins there in the serum, and they do adsorb some of these proteins non-selectively and form what the authors call a protein corona around themselves. And there are actually two components of that corona that they call the hard and the soft corona. So the hard corona is describing the proteins that are close to the nanoparticle, and they have a slow exchange rate with the environment and a thermodynamically favored association with that nanoparticle, as opposed to the soft corona, which is on the outside of the hard corona, and it's more loosely bound and exchanging rapidly with the surrounding environment. And they're able to change the composition of the corona by changing the composition of the nanoparticles as well as their size and shape. And they note that hydrophilic nanoparticles do not adsorb as many proteins from the serum compared to hydrophobic ones. The other thing, and what you're seeing in these graphs on the left as well as on the right here, is that there is a critical temperature that's called the lower critical solution temperature. If you are below that temperature, which is what you see on the top left, you basically have a kind of solvent swollen polymer, and it doesn't have very good interactions with the proteins because the solvent molecules are in the way. And once you get below that LCST, that critical temperature, then the polymer kind of collapses down, and now the proteins can come close and enter the hard corona and have a good interaction. So as with the corona, this temperature at which this transition takes place with these polymers can be adjusted by changing the components. So you can change the feed ratio, the pH, the pKa, how much cross-linker you use. There's several other components that they can change. And they noticed that if they have more hydrophobic monomers in their polymers, that actually makes the lower critical solution temperature lower. So the main conclusion of this is that the selectivity and affinity of these nanoparticles really needs to be thought of as situationally dependent, because depending on things like temperature and pH, you do change the binding properties of them. So now I'm going to talk a little bit about some of the experiments they actually did. And they started out small with a 26 amino acid peptide called melittin, which comes from bee venom, and it is a positively charged molecule that is a membrane damaging toxin, and it lyses red blood cells by inserting into the membrane and creating pores. So this is not going to be interacting with some kind of receptor, and so in order to inhibit it from its action, it's not possible to inhibit a receptor. Instead, the strategy here is to try to sequester the melittin and limit the access that it has to cell membranes. And what you're seeing here is a schematic of how this could work. So the top left there in panel A, the light gray that you're seeing represents hydrophobic interactions. So that is showing a polymer that only has hydrophobic interactions with melittin. And on the right side in panel D, you're seeing the opposite, where it only has electrostatic interactions. And then at the bottom there in B, you're seeing one that has both hydrophobic and electrostatic interactions. So for the very highest affinity nanoparticles that they synthesized, they actually were able to calculate the dissociation constants for these. They calculated it as 7.3 to 25 picomolar. Pico, just to remind everyone, is 10 to the minus 12. And that is actually similar to a natural antibody, which has a dissociation constant of approximately 17 picomolar. Okay, so what you're seeing here in the top part, because the melittin is lysing red blood cells, it can actually be used as a bioassay to test this. And so you're seeing the schematic of how they did this. So basically they incubate it and they do the centrifugation, and you can see on the right side of that schematic, if the nanoparticles are there and optimized, then there's no hemolysis that occurs. The supernatant stays clear. Whereas if you do get hemolysis, you get a red-colored solution. And that's what you see on the right without the nanoparticles. So now looking at the bottom, this is showing results of some actual experiments that they did. Those first two tubes are showing controls, a negative control there. And then you see the first three tubes, 1, 2, and 3. Those are all showing nanoparticles that are not selective for the melittin. And tube 4 is an interesting one because that's actually having accelerated lysis. It's darker red than the other ones, and there's more hemoglobin that's been released because there were positively charged monomers that were incorporated into that nanoparticle, and so it actually enhanced the activity of melittin. And then in tube 6, you see one that they got that actually has a decreased lysis, and that's because they had hydrophobic plus the negatively charged monomers that are able to interact with the melittin by both electrostatic and hydrophobic interactions. So this is only showing the first 7 that they made. There's another one that's number 9 that's similar to number 6. And that was the very best one that they had. I'm going to talk more about nanoparticle number 9 in just a minute. But they were able to measure the melatonin binding capacity of that. It was 180 micromole per gram, which is greater than 10 times the binding capacity of an IgG. So the IgG is on the order, the binding capacity is on the order of 13 micromole per gram. This is showing that in some in vivo experiments in mice. And here what you're seeing is that the nanoparticles are effective in neutralizing melatonin when injected into mice. So on the right side here, this is a 24-hour experiment. And after 24 hours, the controls were all dead. The ones that got positively charged nanoparticles were mostly dead. And I mentioned already before that the positively charged nanoparticles actually accelerate the hemolysis that's caused by melatonin. What I didn't mention in the last slide is that they also increase the toxicity of these. So serendipitously, the negatively charged ones, having negatively charged monomers decreases the toxicity, having positively charged ones increases it. And then if you look at the ones that are both negatively charged and hydrophobic, which is nanoparticle number 9, that's the black line up at the top, that was the one then that survived basically for the entire 24 hours, so that group of mice. They did some toxicity testing in the mice as well. They didn't notice after 2 weeks when they sacrificed the mice any change in their body weight or the histopathology of the organs that they looked at. And they noted that those nanoparticles do accumulate in the liver and kidney, so the liver macrophages do appear to be recognizing them and sequestering them. So as I mentioned, using positively charged nanoparticles is not going to be very effective because those are high toxicity. And so you're not going to be able to use this technique probably against negatively charged proteins. But you definitely can use it for positively charged proteins, and happily a lot of these snake venom components and bee venom components are positively charged because they insert into cell membranes. They were able to calculate the binding capacity again in vivo, and they found that it wasn't quite as good as it was in vitro. So remember, in vitro it was 180 micromole per gram for the NP9, and they found in vivo it was only 30 micromole per gram, which is only about a sixth of that, but it's still twice the binding capacity of the IgG, which is more on the lines of 13 micromole per gram. And they think the difference between the in vitro and in vivo binding capacity has to do with competition from the plasma proteins and biodistribution of the melatonin and the nanoparticles. So now this is going to get a little bit more into the actual work they described in this paper. They started small with the melatonin and they moved up a little bit to a larger molecule, which is phospholipase A2. And this is another component of venom that interacts with lipid membranes and lipoprotein particles. And in the presence of phosphatidylcholine, it catalyzes production of lysophosphatidylcholine, and the lysophosphatidylcholine is what actually induces hemolysis. So in the same way that they were able to do that hemolysis assay with the melatonin, they can do that again with PLA2 as well, since it induces hemolysis. And here they went through a couple of rounds of nanoparticle generation. So the first one that they did, this is showing results from the first group of nanoparticles that they synthesized. You can see in panel A, so the y-axis is showing percent of erythrocytes lysed, and you see that nanoparticle 1-5, which is the fifth one from the left, is the only one out of this group that really did much to prevent lysis. The rest of them really weren't that good. And they were interested in knowing, is it doing it because it's preventing formation of the lysophosphatidylcholine, or is it doing it because it's actually binding to the lysophosphatidylcholine? So in panel B, what you're seeing is they're comparing on the left of those, the solid blue lines, that's showing where you have the lysophosphatidylcholine plus the nanopeptide together, the nanoparticle together, and you see that there's not really a difference if you have the nanoparticle present or not present. And this is nanoparticle 1-5, the one that they know works. You see that it doesn't really matter if it's there with the lysophosphatidylcholine or not. And so they concluded that it's not actually sequestering lysophosphatidylcholine, that what it must be doing is preventing formation of the lysophosphatidylcholine. And then in panel C, what you see is the feed ratio of the monomers that they used. And so I just want to draw your attention that on the far right there, you see they used 2% of that cross-linking. So they then used that NP1-5 as a lead, and they went ahead and synthesized a second round of these nanoparticles. And at the top here, you're seeing the general strategy that they used here. It's similar to what they did before, and same idea, that once they made these nanoparticles, they then went ahead and incubated them and looked to see if there was hemolysis or not. So in this case, they're using bee venom instead of snake venom, and the reason why they did that is because the bee venom contains both PLA2 and melittin. And so these two components, which are both hemolytic, work together synergistically, and this hemolysis assay actually then allows them to analyze for both of their activity at the same time. The other thing they did that was kind of interesting is that they made their requirements here a little bit more stringent. Their testing conditions were more stringent. So instead of only incubating for 15 minutes like they did in the first round, they did this for an hour. And so when they did that, they found that their first nanoparticle, which was the 1-5, couldn't hold up to those conditions, and it was not functional in that case. But when they did their second group, they got one that was actually able to, which was 2-12. And so they went ahead and tested 2-12, and that's what you're seeing down at the bottom in group B, in panel B there. And you see that at 63 microgram per milliliter, they did start getting some effect there for it to be able to inhibit the lysis. And that was specifically for PLA2. And then they also were able to check for the melittin. The melittin, it wasn't quite as good. It was 300 micromolar. If you look in panel C, you can see that here the difference of this is that there's much more cross-linker. It's 15%, whereas it was only 2% for the prior lead compound that they made. So what they concluded is that the amount of cross-linking is significantly going to impact the neutralization of these PLA2s, and even more importantly and impressively, that they were actually able to inhibit two dissimilar venom components with a single nanoparticle. So then the last thing that they were looking at was for selectivity, and here what they, as we mentioned before, when you put these in biological solutions, you do get some adsorption of the biomacromolecules to form those coronas I mentioned before. And so here you're seeing the schematic of what they did, which was to do some incubation centrifugation steps. And then they did analysis with SDS page and LC mass spec. And down in panel B, you're seeing the results of those. So the first three lanes are all controls. The second lane is showing PLA2 by itself, and that's a crate PLA2 from a snake. The lane 3 is showing the serum control with no nanoparticle present. And then in lanes 4 and 5, now they start mixing everything together. So lane 4 has the nanoparticle 2-12 with serum, but there's no PLA2. So in this case, what they're showing is that most of those serum proteins do not adsorb into the hard corona. In lane 5, what you see is that now they've added in some crate PLA2, and you can see that one that's got the green box around it in lane 2, that's where the crate PLA2 is, and you see that that band is also present in lane 5. And lanes 6 and 7 are a similar set of experiments that were done using purified PLA2 from bees, and it basically showed the exact same thing. And then C is again an electron micrograph showing a picture of what these nanoparticles look like. Okay, and then this last slide, this is just a kinetic analysis showing the dissociation of the bee venom nanoparticle and PLA2 complexes. And what they note is that in that green box there's only a small amount of desorption that occurs rapidly, which the authors believe probably comes from the soft corona, and the other 80%, which is in the purple box, desorbs much more slowly. So they were able to analyze the time course of this, and they concluded that the majority of the bound PLA2 that's in the hard corona of these nanoparticles is probably going to stay in there longer than the amount of time that the liver macrophages would be required in order to clear these particle complexes from the blood in vivo. I did speak with Dr. O'Brien, who was a graduate student at the time. He's now since graduated, regarding what's been done since then, because this was published at the end of 2016. So he told me they've been applying these nanoparticles to whole venom extracts, and specifically they've been interested in the 3-finger toxin family in elapid venoms, and they've been doing, again, more in vivo testing in mice. This work has not yet been published, but it has been submitted for publication. These are some references or some of the other articles that these authors published, and there's a lot of other uses potentially for these nanoparticles, so if anyone's interested, some of these review articles that they wrote can get into some of that information. And then I wanted to get an idea, now that you guys have some understanding of the nanoparticles and what their pros and cons are, strengths and weaknesses, how likely you feel that this could potentially be developed. Would the benefits of using these outweigh the risks potentially from them?
So everyone jump on there and weigh in on the poll. All right, we'll close it down. So it looks like 8 out of 12, 67% say yes, and 1 out of 12 says no, and the rest were no answers. So it looks like a bunch of hardy toxicologists are ready to power forward.
Yeah. Well, I'm pretty excited about this myself. I was telling James I kind of feel like I want to go do a postdoc with this guy now and work on developing these some more.
Yeah, it's absolutely fascinating, and you really do wonder what's the toxicity of these compounds. It seems like we need a lot more study, but the potential is fascinating. Okay, and I'll just throw it out there one more time. We're winding down our time here. If anyone has a question, comment, please raise your hand. Not seeing any. Do you folks have any wrap-up comments?
So I think, these antidotes are great, and I think they're all potential, especially, the nanoparticles. There's just so much potential for different venoms. I agree, we don't know the human toxicity, but, only time will tell. If we can make something that's cheap and broadly has a broad affinity, I think we can really make this work. And if Steph ever goes to work for this, I'm going to buy stock in this company. So that's all I'm going to say.
Yeah. This is James and my retirement plan. We're going to buy stock in their company.
Yeah, please fire that info out to the listserv right away. We'd appreciate it.
Yeah. Well, Steve, thanks for allowing us to talk about these articles and having us on.
Yeah, no, actually that was really all the fellowships out there. Those were 3 fabulously presented papers, really interesting, well done. So bravo to all of you. Again, I want to thank you, James, for organizing, Sarah, Kevin, Stephanie, and Paul Waxon. We know you're there. Thanks so much. This was great. Our next National Journal Club will be July 10th, and will be presented by the Pittsburgh group. So we're looking forward to that. And again, thanks so much, guys. This was fabulous.
Thank you.
Thank you. Bye-bye.
Bye.